Okay, great. So let's get started again. The next speaker is Olga Vitek from Northeastern University, and she runs a group there which is focused on statistical analysis of mass spectrometry data and to a large degree proteomics data. And she's also been a long-time participant in these courses, and so we're very grateful to have her here. I also think it's interesting to note that, as a side comment, we always used to have Olga's talks later in the week, like on Wednesday, Thursday, Friday, and then this year we realized that, well, you know, in terms of fundamentals, you know, Olga always says that you need to think about the statistics and the experimental design and talk to the statistician before you start the experiment. So in keeping with that, we're going to have some kind of fundamentals of experimental design and statistical analysis already on Monday. So with that, Olga, please.
Good afternoon. I'm really happy to be here. So as Ben says, I am a statistician by training, and I'm interested in all things quantitative mass spectrometry and proteomics. And so what I would like to do this week is two things. So today we will talk mostly about the fundamental statistical principles of experimental design. So it will be related to mass spectrometry, but the principles are more general than that. And probably many of you are familiar with these concepts, but if you are, please bear with me, because I feel that this is very important for all of us to be on the same page moving forward for the rest of the week. And then I will come back on Wednesday and we'll talk more specifically about the methods and tools that we develop in my lab, specifically for mass spectrometry and specifically for DIA. So we will have an opportunity to do that and also have a hands-on session actually trying things out. But so for the moment, let me talk about the fundamental principles of statistical design and analysis. So the first question is, why do we need to talk about this? Why statistics? And the fact is that in the experiments that we talk about here, variation and uncertainty are a fact of life. So we can really not avoid that. And there are all kinds of variation. There is technical variation, which comes from the way we handle, store, and process samples. Instrumental variation, you know, differences in elution time, ion suppression, and so on. Signal processing, as we will learn later, kind of more and more this week, different decisions we make when locating peaks, quantifying peaks, identifying peaks, they also introduce some uncertainty. All of these things, we do our best to handle that, right? We do our best to minimize the variation and so on. And sometimes people say that, you know, if my experimental workflow is really good and my instruments are really, really good, then I don't need statistics anymore. And I would like to differ, because the most important part here is not any of the instrument and sample storage and data processing components. The most important aspect is that our purpose is to study living systems, right? Living organisms. And there's a substantial amount of variation, natural variation of protein abundance from one person to another, within the same person from one day to another. And so even if our measurement workflows are optimized to perfection, we still have to deal with a substantial biological variation which is present in living organisms. And we need to distinguish changes which are induced by treatments or by stresses or by disease that we're studying from the natural variation which occurs in practice. And so we can think of statistics as a body of methods for making wise decisions in the face of uncertainty. So what does it mean to make wise decisions in this context, and how statisticians can contribute? And I think about this as three components, all of which lead to more effective and more reproducible research. And the first is experimental design, and that's our focus today. How can we design the experiments which are unbiased and efficient? The second is data analysis. And, you know, sometimes people say, well, we need statistics because the data are too large. Well, that's true, but that's not the point. The point is that we want to make sure that our conclusions are objective. They are not affected by our subjective judgment, and that somebody else can take the same procedure, repeat it, and obtain the same results. And so this is why statistical approach to data analysis is really important. And of course, statistical tools, we cannot do all of that by hand. We need to automate that, but it's more than automation. If we write our analysis steps in form of scripts, it also means that they're reproducible. It also means that essentially scripts are the best way to document what we do. And so these three components are contributing to reproducible research, and that's what we'll talk about. And so before I start, I would like to first talk about how we can translate the objective of a study into a specific statistical question. And the reason why I like to talk about that is because oftentimes people come to me and say, I have this really great samples, I just want to see what happens. Well, guess what? If you don't have a specific question, nothing happens. I can tell you this right away. And so oftentimes we need to think before we start even doing anything is to say, okay, what is my scientific, biological, clinical question? And then how can I translate this into a more specific statistical or machine learning question? And this will allow us to move on in terms of experimental design, data analysis, and so on. So I will use as a kind of context here discovery of biomarkers because it's very easy to think about, but you can kind of project this to other problems in fundamental science and so on. So we can think of statistical goals. We can kind of broadly classify them into three groups. So the first statistical goal is class discovery, where we say, okay, we want to find patterns in the data, and we don't really have any particular definition of a pattern. We just want to look for things which are similar to each other. So an example would be a heat map like that. You have all seen this. Here the rows will be proteins, the columns will be biological samples, maybe time points, and the color will be the abundance of a protein in each sample. And we would want to say things such as, okay, let's cluster proteins which have similar patterns across samples. And this is meaningful because maybe these proteins are involved in the same biological process. Or let's cluster samples in the space of proteins. And this is also meaningful. Maybe these are subtypes of a disease or something like that. So both things can be useful. The important thing is we really don't say what type of pattern we're looking for. And this is why this is called an unsupervised method, meaning that we don't tell the method what we're interested in. We just say, go and find things that are similar. And other examples would be principal component analysis. Here, the axes are linear combinations of protein abundances. The points are the samples, and if they fall together in the cloud, it means that they have similar patterns of protein abundance, or looking for kind of profiles like this. The important thing is that there is no labels of classes such as healthy and disease. Therefore, we cannot think of these methods, for example, as tools for discovery of biomarkers, or as methods to see what is changing and what is not changing, because the method doesn't have a sense of what is useful. And for example, plots like that, they're really good to diagnose problems with quality, so for quality control. However, and I see this sometimes in the papers, people say, I have disease groups which cluster in PCA plot, therefore I found biomarkers of disease. This is wrong because this method has no concept of disease, right? We didn't give this information. And therefore there's no concept such as error rates, accuracy, and so on. So these methods are best used for data visualization, data exploration, QC, and so on. But that's really the limitation of these methods. The second statistical goal is class comparison. Now, this is a supervised method because now we have a concept of a class. So we say, okay, these are healthy subjects, these are subjects with a disease. So the method knows which is which, right? And so here, if we take, for example, one protein, let's say the y-axis is the abundance of the protein on the log scale. Here we have replicates from the healthy group. Here are the replicates from the disease group. We want to know if on average between healthy and disease, the protein is upregulated or downregulated. And this is a very common question which we ask in mass spectrometry. And the methods would be t-test, analysis of variance, and the readouts would be p-value, posterior probability maybe, and things like that. So this is a very common task. What I see in papers, often people say, okay, I have a protein with a small p-value, therefore I found a biomarker. And again, this is wrong. So proteins with small p-values are not biomarkers. And the reason for this is more subtle, because we need to first define what do we mean by a biomarker of disease, right? And biomarker is something which is supposed to be actionable in clinic, right? So a patient comes to the doctor and the doctor needs to decide, okay, this or this treatment, right, and so on. This method compares averages over multiple subjects. It wants to know if the mean of these subjects is systematically different from the mean of these subjects. The doctor doesn't see an average patient. The doctor sees an individual patient. Therefore, methods such as t-test, ANOVA, and so on are not appropriate for discovery of biomarkers of disease. They're nice for initial screening, they're nice for some problems in the basic science and so on. However, these are their limitations. So let me just give you a little bit more insight into why differentially abundant proteins are not necessarily actionable. So here's an example. So we have log of the abundance of the protein. These are replicates. They're tightly clustered around the mean of this group versus this. So very clearly, if you have a subject with this protein abundance, it's likely to come from the disease group. This is fine, right? But now here we have a different situation. Same change in protein abundance between the groups, but a lot of natural biological variation between the subjects. So if someone comes with this protein abundance, it can be both. So this is not actionable. However, we may be really, really confident in this mean, really, really confident in this mean, and we can claim that this change is not an artifact. So this protein will likely have a small p-value, but will probably not be actionable. And here's another kind of the opposite case. So here we have two proteins, protein 1 and 2. Each point is a person. Let's say those are healthy and those are disease. So here is very clear that disease subjects have upregulated protein 1 and 2, right? So they're both changing and actionable. But in this case, we see that protein 1 spans the full range of abundance for both healthy and disease. And protein 2 also spans the full range of abundance for healthy and disease. So neither protein 1 nor protein 2 will have small p-values in this case, because both have the same ranges, right? However, as a bivariate panel of two proteins, they're perfectly predictive, so they're perfectly actionable. So these are, so essentially because of that, because the objective is different, we need different methods when we focus on biomarker discovery. And so for that, we need this statistical goal 3, which now focuses not on comparing the means, but predicting the status of individual data point. And this type of methods will be machine learning methods such as logistic regression, random forest, support vector machine, deep learning, whatever you would like. And the readouts would be sensitivity, specificity, precision, recall, positive predictive value, things like that. And so the example would be here, we have two proteins. So essentially these methods all look for a boundary which separates one group from the other. And again, it's a supervised method because the method knows from the example data, right? So this is example of healthy and this is example of disease. So here the decision boundary ignores protein 1 and just says, well, large values of protein 2 are healthy, low values of protein 2 are disease. And it works okay, but clearly some of the red points are misclassified. So here we can have a more complex boundary which uses both protein 1 and protein 2 and does a better job. So now we can probably have a better predictive ability from this method. And now we have a method which is really, really complicated, accommodates really, really well each data point in our example training set, but it's likely too specific and accommodates too well the artifacts in our data. So this will likely be an overfitting and it's likely not be reproducible later on. And so the goal of this method essentially is to look for boundaries which are kind of in between too simple and too complex and find ones that are just enough. So my point is that before we even start, we first need to decide, okay, what are we doing? Are we interested in just looking for patterns? Are we interested in comparing averages? Or are we interested in predicting the status of individual samples? And this will, you can see already, will direct us to very different methods. And if we have different methods and different objectives in mind, it means that it will have different implications for experimental design. And so these are really important things to consider before we even start doing other things. And of course, if we work, again, going back to the biomarker discovery, if we really are in the business of discovering biomarkers and advocating for them, this is what we have to do. That's really the standard. Okay, so how does it sound? Do you have any questions? Okay, well, don't hesitate to interrupt me because I can talk a lot. Okay, so now let's go to the experimental design, right? So for the rest of today, and in fact, most of today, and in fact, next Wednesday as well, we will focus on the comparison of means. So hypothesis testing, comparison, looking for proteins which are differentially regulated, because this is one of the common goals in many experiments. So let's kind of stay within this goal 2, right? So looking for differences in abundances on average over all the subjects. Okay, so if someone decides that this is our objective and we need to design an experiment, I'd like to show you now what's going on in my mind when I hear about the problem, because what I try to do is to project this into the abstractions which use statistical concepts. So let's assume again that we have two populations, healthy and disease, and these two populations, they are infinitely large for all practical purposes, and they are identical in terms of everything that can affect protein abundance, such as age, gender, lifestyle, and so on, except for the fact that this population is healthy and this population has a disease. And we want to know which proteins are differentially regulated or differentially abundant between healthy and disease. So the population is large, so we cannot interrogate every single person in this population. It's not possible. So what we will do, we'll include in our study a subset of the subjects from each population. And so this is our first layer of randomness, because if we include the individuals from the study today and want to repeat this study tomorrow, we will not have the same individuals. So we will not have the same protein abundances in our investigation. So this is the first layer of variation, and this is the biological variation, because there is a natural person-to-person variation in protein abundance. But this is not the whole story, because even if we include the subjects in the study, we still don't know what their protein abundances are. We need to actually measure them using the technology, which for the moment is subject to noise and uncertainty and variation. So what we actually see is the measurements from these subjects. And this is the second layer of variation, and this is the technical variation, which reflects uncertainty into what is in the actual biological samples. And so the goal of statistical inference is to take what we see, because the only observable thing is actually this, and make the conclusions about what we don't see, and what we're interested in is this. So we want to make the conclusions about the unknown. So statistical inference is to take the data, what we observe, and make statements about what we don't see in the entire underlying populations. And this has two problems potentially. The first is bias, meaning that our conclusions about the unknowns are systematically wrong. And bias is the worst kind of problem, because bias is reproducible. So if we have some problems or deficiencies in the experimental design, we will repeat the same problem again, and we will have the same conclusions, and we will say, great, we have reproducible results. Well, we're reproducibly wrong. So the first goal of statistical experimental design is to eliminate the bias. And the second goal is eliminate inefficiency. And this means that the data that we actually observe are so noisy that for all practical purposes, we cannot say anything useful about the underlying populations. And so the second goal is to design the experiments to make sure that the level of noise is low enough that we can actually make meaningful statements about the populations. So this is really the objective, and this is how statisticians think about the experimental design. I will do minimal math. I will not do many formulas, but let me just show you how statisticians think in abstract ways. So the convention is that we use Greek letters for everything that we don't see, and we use Latin letters for things that we actually see. And so we assume that there are mean abundances of proteins in the underlying population, and they exist out there. They're numbers, they're fixed. We will just never, ever know what they are. But we assume that they exist and they have fixed values. And then there are also abundances of proteins in individual subjects, which also exist, but we do not know them. What we actually see is the quantified peak intensities in DIA experiments or in any other experiment. And so now our goal will be to take some summaries of the observed peak intensities. I will tell you upfront, we like to work with log intensities. I will come back to this a little bit later. And so the summary will be on the log intensity scale. And then we would want to use this to make statements about the difference between the means and the populations. And so bias in this case means that this difference is systematically different from this, and inefficiency means that the variance of the summary is too large. And so statistical experimental design will attempt to minimize these two quantities, these two discrepancies. Okay, so how do we do it? So the bias and inefficiency can be prevented by 3 fundamental principles of experimental design, and I did not invent them. And we didn't invent them for mass spectrometry. They have been around for like over a century. They have been developed by scientists such as Ronald Fisher earlier, like in the case of agricultural experiments. But interestingly, every time there's new technology comes about, somehow people think that these principles do not apply anymore. They apply very well, and we need to make sure that we continue to use them. So let's see, the first fundamental principle is replication. I hope I don't need to convince you now that we need to have replicates. But let's just kind of stay with that and think about why replicates are important. In fact, what I would like to do is throughout the discussion today, I would like to come back to the issue of replication and sample size and why it is important. Well, so let's have a very simple experiment. So we have a label-free, let's say, DIA experiment, and let's say we have 1 feature per subject, just to make it simple, right? So 1 feature per protein per subject. So each protein is represented just with 1 peak. And so now we have only 1 subject. And so we have a value for healthy and value for disease. Is it systematic or is it an artifact? Exactly, right? Who knows, right? Because we have no idea of what artifacts look like, right, in this, and what systematic change looks like. So these are 2 situations as we have seen before. So now we have multiple subjects, and they're tightly clustered, so this change is not likely to be an artifact. Here we have exactly the same difference of log abundance, but there's a lot of variability, so this is more likely to be an artifact. So replicates allow us to do 2 things. One is quantify how much uncertainty we have in the data and to distinguish artifacts and not artifacts. And the second is, well, if we have more and more replicates, we will be more and more confident in these means, and we will be able to make statements that they're systematic or not systematic. And by the way, the consequence of this plot is that if we only look at fold changes, and we'll use fold changes to make cutoffs for differential abundance, it makes no sense whatsoever, right? Because exactly the same difference, exactly the same fold change cutoff may or may not be an artifact depending on how much variation we have. And so since now we need to have replicates and we have 2 sources of variation, biological and technical variation, well, we have 2 types of replicates. We have biological replicates and technical replicates. We will talk about this more, but let me give you an upshot. Biological replication is most important. If you have a fixed number of runs in your budget, you will be much better off to use each run for a new person every time, as opposed to take 1 person, quantify this person really, really, really well with many technical replicates, but it's still 1 person, because we still don't know how much natural biological variation we have in the population. So this is most important. Okay, the second fundamental principle is randomization. And this, you know, I also hope that you are familiar with that, but I'm surprised how often it is overlooked. And I really see this in my practice, just in collaborations, but also as a reviewer. This really remains a problem in our community. And so it's really important for us to remember that. So let me give you an illustration of why it's important. So again, let's say we have 1 peak per protein per subject to make it simple. And so let's say we have multiple healthy subjects. They're all acquired on day 1 and day 2. And then we have disease subjects which are all acquired on day 3 and day 4. And so now we have a difference which is likely to be systematic, except that this difference can be due to 2 sources. It can be due to the disease, but it can also be due to day of data collection. So what do you think is the most likely source? Day of data collection. And day is usually a proxy for many things, right? Changes in, you know, operator, temperature, batch materials, and whatnot. So this would be an example of an experiment which is perfectly confounded between the disease group and time of data collection, and it's essentially wasted time and money, because there's no amount of statistical sophistication which will be able to deconvolute these 2 sources completely. So instead, what we can do, we can randomly assign each subject in each group to day of data collection. So now we'll have a situation like that where we have more variability around the means, because now the variability comes from two sources, the biological variation and the day-to-day variation, which adds to that. But at least we don't have a bias, right? So this will be systematic bias. Now here we have somewhat inefficient experiment because we have more noise, but at least we don't have the bias. And especially when the sample size increases, roughly the disease groups are equally allocated between all of the days, and so this is eliminated. And because we have two types of variation, we also have two types of randomization, right? We need to randomize the selection of subjects when we select them from the population, and we also need to randomize samples at all steps of data processing and spectral acquisition. So this is really important. So let me just give you some examples. So this is one old publication, but I really appreciate that. This was by statisticians from MD Anderson Cancer Research Center who do a lot of collaborative work and with design and analysis of data for clinical researchers. And they published a paper essentially with examples of failed experiments. And I think it's very insightful. It is old, so they used old technologies, cell it off, it doesn't matter. So here they received samples from 5 types of cancer, and it was blinded, so they didn't know which is which. They did all the right things in terms of peak picking, normalization, and so on. They did the unsupervised clustering. They found 6 groups. And they were excited, thinking, oh, we found a new cancer subtype. But what they did then was they have a plot here where the x-axis is the order of the samples as in the clustering, but the y-axis is the date of data collection. And you see very well that each group corresponds perfectly to the date of data collection. And turns out that the beginning they had some cancer types, and then at the end they had some other cancer types. So this is wasted time and money very clearly, because we cannot take these things apart. But it can be worse. So here's an example from a publication which is actually not even that old, although somewhat old already. It's 2007. And it is also a very kind of very interesting case. So here it's coming from genomics. So people studied gene expression and difference between gene expression between Asians and Europeans. And what they found is that 78% of genes were differentially expressed between Asians and Europeans. 78% of genes were differentially expressed. You know, we're not that different, right? So what happened was, and you know, the good thing is, so there was all the data was there, the analysis was there, and so this comes from Jeff Lake, who is a biostatistician at Johns Hopkins, and his colleagues wanted to reproduce that, and so they asked if there was any flaws in the design. So they went through all the scripts and and they didn't find any problems, so all was fine, right? So the statistical analysis was all right. Luckily at that time, so this was using Affymetrix microarrays, and the raw data were available. And so at that time, Affy chips had a timestamp in the corner of an Affy array. And so what they realized at that level, they had to go that far to the very raw data to realize that there was a 1-year difference between collecting the data for Asians and for Europeans. No wonder things are different, right? But they're different for wrong reasons. So it has nothing to do with what the researchers studied. So these things occur up to the date, and so we really want to avoid this as much as possible. Okay, and this is like one of the famous quotes from Ronald Fisher: to call in the statistician after the experiment is done may be no more than ask to perform a post-mortem examination. He may be able to say what the experiment died of. Therefore, as Ben was mentioning, what I would like to make sure you know, if everything else you forget, right, and you don't remember, it's too complicated, the biggest message that I have for you today is talk to a statistician before collecting the data. This is the most important part, because once we have flaws of design, nothing can be done. You know, if the analysis was wrong, you can go back and reanalyze the data, right? But with the design, it's really wasted resources and time. So it is really important. Okay, so this was randomization and replication and randomization. Oops. And the last principle is probably the most technically challenging. And so I think replication and randomization you can do yourself. Blocking is the principle which really requires more statistical expertise. But it is really effective. And the goal is to reduce both bias and inefficiency. So here's going back to our experiment with the previous study. So if we randomize completely, so now we have more variability in each group because it has the biological variation and the day-to-day variation added to that, right? If we know that day-to-day variation exists, we cannot do much about this, it's a fact of life, but we know that it's present. What we can do is to impose a restriction on randomization. So essentially blocking is a restriction on randomization. What we can do is to say, on day one, let's restrict us to have one healthy and one disease on the first day. There's still randomization. I have one randomly selected healthy and one randomly selected disease, but one of each. Then day two, let me have one of each as well. Day three, and so on. And so now I have the extra information that these two measurements were all collected on day one, these all on day two, and so on. And what I can do now is to look for differences between healthy and disease within each day. Again, remember difference on the log scale, right? So it is the same as looking at the log fold change. And so instead of comparing the mean of these points to the mean of these points, I'm looking at the differences between pairs. And you see that the differences here all point to the same direction. And so this indicates that we have an upregulation in the disease. And so this requires two things. This requires us to allocate the samples to the data collection steps in this way, but also requires us to analyze the data accordingly, so manipulate the data differently. And so this is why you need to design the experiment in a way that you know how it will be analyzed later. And here I'm using a pair within each day just for illustration, but there are statistical techniques which generalize this. What if you can do 10 samples a day, right? So how would you block on 10 samples? What if you have plates and then also some other kind of groupings? And we can kind of work with that. So this is a more involved type of steps. But again, because we have biological and technical variation, we need to block on biological replicates and on technical replicates. And so the benefits are that so, you know, if you have a small sample size, just accidentally you can still end up, even if you randomize, you can still end up with more healthy on first days and more disease on the last days. So blocking ensures that it's perfectly balanced. And we also essentially eliminate the day-to-day variation by looking at the pairs, so it gains efficiency. You know, sometimes people also, I heard somebody telling me, I don't like to work with statisticians because they ask for replicates. It's true, and we'll talk quite a bit about that, but the point here is that we can get out more of our data with a fixed sample size if we allocate the resources appropriately and if we analyze the data appropriately. So it's not all about just having more replicates, you know, it's also how to allocate our resources and analyze the data in a smart way. So just a few examples. So here's again from this paper from MD Anderson. Here it was another study with two types of cancer, same thing. They did all the right things. They found two groups. They were happy only to realize, well, after they unblinded, that there was a protocol change in the middle of the experiment. And again, these were all one group and this was another group. So again, wasted experiment. Protocol change may be unavoidable, especially if we have long running studies, right, with many replicates, because we need to change a batch of materials, we need to do some maintenance, you know, we need to have a operator changed and so on. What we could, blocking in this case would mean that we have roughly the same number of healthy and disease before the change and roughly the same number of healthy and disease after the change. And then we will look for differences here and then integrate the differences from the two batches. This would be a much better way of doing it. And also to illustrate blocking with respect to biological factors, when we talk about biological factors, blocking is sometimes referred to as matching. And so just to illustrate that, so if we compare healthy and disease, as I mentioned before, so we assume that the confounding factors such as age, gender, you know, lifestyle, and so on are roughly equally distributed between the two populations. However, when we include subjects in the study, and especially when the sample size is not very large, just by random artifact, we may still end up you know, with healthy, which are all male and older, and then disease, which are all female and younger, just by random chance. And so blocking will again impose a restriction on randomization, and will say, okay, I want to have one female from a certain age group from each population. You randomly select this type of person in the study. And then you say, I want to have one older male from, or older female from each disease group, and you randomly select one person from each group for this study. This way we ensure that these confounding factors are equally distributed between healthy and disease group, and they do not introduce confounding. So this is an example of a collaboration that we had a long time ago with Munich Heart Center. So this was a group that was interested in heart disease, and they were storing blood samples from everybody who came to their center for any problem. And so they had essentially a biobank with a large number of those samples, and they wanted to include a subset into their investigation. And so this was just kind of part of that study. So the clinicians think that age and gender affect pretty much everything, so they wanted to make sure that we stratify or block with respect to age and gender. So they came up with these 4 groups: older female, older male, younger female, and younger male. These were the numbers in their repository at that time. And so if they wanted to have an initial exploratory experiment, so the goal would be to have the same number across all disease groups for this age and gender, the same number for this age and gender, and so on. So essentially, the rows have to have the same numbers. Between different rows, you don't have to have the same numbers, but within each row, between disease groups, you want to have the same number. Therefore, if we now compare control, for example, with this disease group, we know that the age and gender groups are equally represented in both disease groups. So that's the idea for that. Any comments?
If you use spiking standards, can we minimize randomization or replication?
We will get to that on Wednesday, but let me give you the upshot, not really. And the reason is that every normalization makes assumptions. For example, if you have a single standard and you essentially equalize a single standard, you assume that every protein is affected by whatever artifact exactly the same and identical way as your standard. And in most cases, especially in this kind of large-scale DIA experiments, it's a very strong assumption. And so what happens is that at some point, your results reflect your assumptions a lot more than they reflect your data. And this is not a good place to be. So you're much better off to design an experiment in a way that does not require this type of assumptions. And we had quite a bit of experience also with controlled samples and samples where we have some sense of biology, we know what's going on, and when our collaborators forgot to randomize, and essentially I had to push back and say, we cannot be involved in this project unless you go back and you redo it and randomize, and they did, and we were struck by how different the results were, like before and after randomization, even if you do normalization and so on. So don't count on that, because there's always a lot more going on that you think, and then you can eliminate with the standards. Yeah.
How to determine the minimum sample size to get the accurate information?
Hold on to that thought, we will get to that. It's actually a very complicated question, and I haven't said enough yet to answer that. But to give you an upshot, the minimal sample size depends very much on what you do. And it depends on the complexity of your biological system, it depends on the proteome coverage, it depends on the technological considerations and so on. Meaning that your sample size will be very different from somebody else's sample size for whatever study. So I cannot tell you just use 3 patients, no such thing, right? And I hope you will appreciate that by the end of today and Wednesday as well. Okay, so, but to determine sample size and determine experimental design, we need to know how we're going to analyze the data. And so usually the way we do it is we talk about data analysis, and this is why, what I will do next, I will talk about the t-test. And I know that many of you probably have seen it, but again, let's just do that so that we can see how this translates itself later into sample size and other considerations. So, 2 groups, and I will just talk about comparing the means between the 2 groups and talking about t-test and p-values and so on. And what I will do first, I will tell you about how good the p-values are, and then I will tell you how bad p-values are, right? And so we will kind of look at it from both sides. Okay, going back to the kind of abstractions which go on in my mind when we talk about 2 group comparisons, right? So we try to summarize what we see, right? And make statements about what we do not see, but what we're interested in, right? And we have to deal with bias and variance of our summaries. Again, let's make it simple. I will tell you on Wednesday how to deal with many peptides and many fragments, you know, and so on. For today, let's just say we have 1 peak per protein per person, just to kind of keep it simple. And our goal is to compare the means between the 2 groups, healthy and disease. So now let me just kind of take a pause here and clarify why I always bring up log abundance and why we work on the log scale. So first of all, when we do that, we so the log measurements, they have much better statistical properties in general. So however, if you come especially from analytical chemistry, you know, or from this type of background, you probably are very used to talk in terms of the fold changes and ratios, right, and comparison of things. So the fold changes on the log scale have log fold changes have a clear relationship to the fold change on the original scale. So if we want to compare 2 groups, the fold change between the groups will be some kind of typical or consensus or representative value in group 1 divided by some typical or consensus value in group 2. Now, if I take the log of this, it will be the log, you know, log of the ratio is the difference between the logs, right? So log of the fold change is the difference between the typical value in group 1 and typical value in group 2. So now what happens if we take the log of the peak intensities and, for example, have the mean of the log intensities in group 1 minus mean of the log intensities in group 2? So I will just spell it out. So the mean of log intensities in group 1 is just, well, take the sum of all the log intensities, divide by the total, right? Okay. Now, I will actually explicitly mention that y is the log of the original values. So now log x is the original values of peaks, right? Peak abundances. So now I have the mean of the log abundance in the first group minus the mean of a log abundance in the second group. And now log has this property that if we take the sum of the logs, it's the same as the log of the product. So you can rewrite this like this. And again, difference between the logs is the log of a ratio. So if this is too obscure for you, don't worry. Essentially, the upshot here is that when we work with log intensities of peaks, our estimates of fold changes have the typical value in group 1, which is the geometric mean of the peak intensities on the original scale, and typical value in group 2 is the geometric mean of the log intensities on the original scale in group 2. So from now on, I will talk about log intensities of peaks and log fold changes, but you can think of this as the log of the ratios of typical values, where typical value is the geometric mean on the original scale. Okay, so this is an aside. So now let's talk exactly why we want to work on the log scale. So thinking about changes in abundance between the 2 groups, we have these concepts, right, that we have the constant or the mean protein abundance in group 1 and the mean protein abundance in group 2. So these are Greek letters. They are unknown. We will never know what they are, but hey, we can make guesses. So let's make a guess. So let's state that our null hypothesis or our assumption is that in the underlying population, there is no difference between the 2 groups. So the protein is not differentially regulated. The 2 means are the same in the population, right? Not in the data. And usually the null hypothesis is something we're not really interested in. This is kind of the status quo, right? And the alternative will be something we are actually interested in, and it is the fact that the difference between the two means in the population is something that is not zero. And so now what we will need to do is to construct essentially a signal-to-noise ratio. So our signal will be the difference between the two group means, right? So this is essentially our log fold change, right? Here we have the means of log intensities in each group. And the denominator is the noise associated with the signal. So let me spell it out a little bit more. So here, those quantities, these are just sample variance in group 1, right? And this will be sample variance in group 2, divided by the sample size in each group. So this is sample variance divided by the sample size. So this is the variation associated with the mean in the first group. Taking the square root of this is the standard error of the first mean. So the standard error is different from the standard deviation, which would be the square root of this. So before we get kind of more confused, let me give you an example, and we will come back to this. So here's what's going on, essentially. We want to know how much uncertainty we have in the individual mean, right, of the log intensities. Here's an example from a whole collection of vignettes published in Nature Methods. So it's called Points of Significance series, and it's a collection of like small 2-page notes on different topics in statistics. And I have a lot of figures from this series because there is text that comes with them. And if you want to know more, you can go and you can actually find the text. So let's talk about what it means to have uncertainty in the mean. So if we start with the data which have normal distribution, so roughly bell-shaped. So this will be the abundance of one protein across all the subjects in the population, if we could somehow characterize this. What we're actually doing is we're taking, let's say, 3 subjects from this population, calculate the mean, go back, take 3 more subjects from the population, calculate the mean, 3 subjects from the population, calculate the mean. This is the histogram of those means of 3 subjects after we repeat this many times. So what we see is that this histogram, well, it has some shape in it, right? It's roughly bell-shaped. It's narrower than the histogram associated with the original population. And this is because the means of 3 subjects are more stable than just one value of one subject, right? Now, if we do this again with 5 subjects or 10 or 20, we see that the histogram of the means becomes narrower and narrower, because the means are more and more reproducible if we increase the sample size. So these histograms here are called sampling distribution of the mean. Sampling refers to the fact that if we sample 10 patients have the mean, if we go back, sample 10 more patients have the mean, how much variation we will have between the means. And so we see that as the sample size increases, we have less and less variation, meaning that our means are more and more similar to each other if we repeat the experiment. They're more and more reproducible. And so this is roughly bell-shaped, and the variance of this histogram is this quantity. So this is the variance of the sampling distribution of the first mean. And you see that sample size is in denominator. So as n increases, the variance becomes smaller and smaller. Right? And so the square root of this is the standard error. So it's just essentially the standard deviation of the histogram of the means. So a really important distinction is the standard deviation versus standard error. Standard deviation is the variability in the original population. Standard error is the variability in these histograms. And so standard error is what we need to characterize the uncertainty in our summaries, because the summary is the mean of multiple replicates. Okay, so this is good. Now, what happens if our data are not normal? What if they come from some kind of weird distributions? And this is, by the way, something that people often ask me, oh, but my mass spectrometry data are not normal. Is it a problem, right? So do we need to do something else? So let's take a look. So we start from these different distributions, which have all kinds of crazy shapes. But if we look at the sampling distribution of the mean, it becomes increasingly bell-shaped. And it becomes increasingly narrower. And this is what this type of behavior is what we call the central limit theorem. So as the sample size increases, the means look more and more normal and more stable, even if we start from distributions which are somewhat crazy. And therefore, since we are interested in means and not in individual values, actually assuming normal distribution is often okay. However, you see that if we start with something which is very different from normal, we see that this histogram is wider than this histogram. And it takes essentially larger sample size for this to look like a normal distribution. Therefore, we'd like to work on the log scale, because on the log scale, our data look a lot closer to normal than on the original scale. That's really the reason for the log transformation. Okay, so now what we do is going back. So we build the signal-to-noise ratio, right? So this was our signal, the difference between the summaries. Now we know that these means on the log scale kind of come from more roughly bell-shaped distributed kind of population. This is the uncertainty in the first mean. This is uncertainty in the second mean. We take the square root to bring it on the same scale. So this is the signal, and this is the noise. So this is what we do in terms of kind of asking ourselves, does the signal exceed the noise, or is it more than the noise, or is it less than the noise? Okay, let me skip that. So now, how do we know that how large is large? So how large should the signal be to call it systematic as opposed to an artifact of random chance? And so this is where the assumption of normal distribution is actually very helpful. And again, it's either exactly normal if our data are normal, or the means have approximately normal distribution by the virtue of central limit theorem. So the normal, if we can assume the normal distribution, and if we assume that each subject is independent, if in fact there is no difference between the groups, so if those means are the same, then we can predict theoretically, without any data, just mathematically, analytically, what would be the plausible or possible range of those signal-to-noise ratios. And this range, or rather the distribution of the signal-to-noise ratios, looks like that. If the data are exactly normal, it is exactly student distribution. If it's approximately normal, then it's approximately normal distribution. Essentially, we can predict mathematically what is the shape of this distribution is. And this is very valuable because it tells us what is the expected range of the signal-to-noise if there is no difference between the groups. So now what we can do is to actually calculate this difference from our data and overlay this on top of this distribution. So if it falls roughly in the middle, then we say, yeah, well, that's what we expect, right? So there's no evidence against our assumption. However, if it falls in the tails, then we say, wait, that's too unusual. Therefore, something is wrong. The first thing that is wrong is that we assume that there is no difference between the groups. So this is evidence against the null hypothesis in favor of the alternative. And so the large piece is we need to establish some kind of goalposts and say, okay, so what is the threshold when we say it's in the tail versus it's in the middle? And usually we select the value of alpha, which is the probability of finding differences when in fact there is no difference, so probability of false positive. And the property of this distribution is that the area under the curve is the probability that the event occurs. So we will have, let's say, alpha is 5%, so half of this goes to the left, half of it goes to the right. And so now if we overlay this quantity from the data and it falls in this rejection region, we reject the null hypothesis, right? Otherwise, we fail to reject. By the way, this is a very important language, so if we don't find, if it falls here, it still doesn't mean that our hypothesis is wrong or true, so we just say that we don't have enough evidence to reject the null hypothesis. And the very last piece here, which is the kind of related quantity, is the p-value. So this is just another way to look at the same thing. So now, because usually we don't know if we expect regulation in this direction or in this direction, so we actually overlay this quantity on top of this curve left and right, and the p-value will be, so the symmetrically, and the p-value will be the area under the curve to the left and to the right of the observed values. And so then if the p-value is less than alpha, it's the same as to say our signal to noise is in the rejection region, so we reject the null hypothesis. If the p-value is larger than alpha, so larger than 0.05 or so, then we say there is no evidence against the null hypothesis. That's it. Right, so that's really the hypothesis testing with with the two groups and with the t-test. Any questions on that? Okay, so the hypothesis testing sometimes can be, well, it's useful when we have many, many proteins and we can just list p-values next to each protein ID and so on. But the p-values do not really tell us the direction of the change, and they do not tell us how much uncertainty was in the data. So information about the signal and about the noise is kind of lost. So another really good way of reporting the results when we compare two groups is in form of error bars. And error bars kind of characterize both the signal and the noise in our data. We already talked about two ways of characterizing the variability. One is the standard deviation in the population, right? And the other is standard error of the mean. So how much variability is in the mean of the data, right? And so here we have the y-axis is the sample size, the dark intervals, let's say the gray interval here is the standard error of the mean. So the standard error of the mean will decrease, right? And of course, there's also the standard deviation, which is not shown here. But the last thing, which I haven't talked about yet, is the confidence interval. So essentially, there are three types of error bars, right? So standard deviation, standard error of the mean, which is different, right? And the confidence interval. And the confidence interval can improve upon the error bars of standard errors and standard deviations because it has a probabilistic interpretation. So confidence interval, it's actually not an interval, it's a procedure, such that if you repeat collecting the data and applying the same procedure over and over, and let's say we have 95% confidence interval, 95% of times the confidence interval will include the true value, and 5% of times will not include the true value. So let's see, for example, here, if we have just one population and we have the population with this mean, if we keep collecting the data and using this procedure, so 95% of times the interval will cover the true mean, and 5% of times the interval will miss the true mean. Now, how do we know that this procedure has this property? Kind of exactly by the same argument as here. If we can assume normal distribution and independence, we can mathematically derive this procedure. And this procedure actually looks very much as before. So this is our signal, this is our noise, and this is the multiplier, which is the value, the goalpost that we have here, and the goalpost that we can have here. So it comes from exactly the same mathematical argument as in the hypothesis testing. So once again, for one mean, right, it would mean that it covers the true value 95% of times. Here we compare two means. This procedure, if we keep collecting the data, the interval will cover the difference between the two means 95% of times. And so you can see already, again, that the sample size is in denominator. Therefore, as we have more biological replicates, the interval will be narrow and narrow. And so here the dark line is 95% confidence interval. You see that it becomes more narrow as sample size increases. However, it's wider than the mean plus minus standard error. And so this is really an important point because oftentimes people get very impressed by the width of the error bars. And what I would like to emphasize now that since we have 3 error bars, they can look very differently depending on what these error bars are. And we need to be very careful when reporting that and not being too impressed with narrow error bars because they can be made kind of artificially narrow. So let me just illustrate that. Also from the same vignettes. So here they always take 10 replicates and they look at the 3 error bars, standard deviation, standard error of the mean,
It's
and confidence.
8 o'clock.
Sorry about that. So 10 replicates, and they have 3 error bars which look identical, except that this one is standard deviation, standard error of the mean, and confidence interval. And you see how different p-values are, so they really reflect very different type of information in the populations, right? But they look identical. Here's the opposite. They keep p-values fixed, and they show how different the error bars would be to give us the same p-value. And you see that the standard deviation can actually have a lot of overlap and can tolerate a lot. Standard error of the mean has to be really far apart with sample size 10. And confidence interval can actually have some overlap. So the message is, first of all, when you report error bars, make sure you say what they are, because they have very different interpretation depending on what they do. And second is, don't be too impressed with error bars which are too far apart or having much overlap, because again, it depends on what they represent. And here it shows how the kind of for different p-values, what the standard error looks like versus what confidence interval looks like. So you see that, especially for larger p-values, you can have quite a bit of overlap in confidence intervals. Okay, so another important concept is statistical power. And this is kind of going closer towards sample size calculation, right? Because, okay, what we have done so far is we said we assume that there is no difference between the groups. We build signal to noise. Now we see how far the signal to noise is from the typical region or range if, in fact, there is no difference between the groups. Okay, how can we now influence our ability to actually see the difference, right? So now, what are the things we can vary? What are the things we can act upon to actually improve our way to detect the changes? And this is kind of thinking about experimental design. And so the concept of statistical power is the concept of how likely are we to actually reject the null hypothesis when it is not true. So let's go back to the signal to noise. Let's imagine that we know that the group 1 is expected to be upregulated. So let's say we have alpha only in one tail, just for simplicity. So this is the distribution that we expect if the null hypothesis is true. So in this rejection region, we put all alpha on one tail. This is 5% false positive rate. So this is also called type 1 error. And this distribution is the distribution of the signal to noise when, in fact, there is no difference between the groups. Okay. But what if there is a difference between the groups? So turns out that we can use kind of similar mathematical argument and also say, well, we can actually predict what the distribution would look like if, in fact, in group 1, the protein is upregulated. And so it turns out that mathematically we can see that, well, if group 1 is upregulated, it means that this signal to noise ratio will be systematically large and away from 0 in the positive domain, right? And so this distribution will be shifted. And by how much it's shifted, well, it depends on how much the mean of the first group is different from the second. And so now, if we reject the null hypothesis every time the signal to noise ratio in this region, well, it's actually in this region with this probability because the true distribution is shifted, true distribution is away from 0. And so statistical power is the area under the curve, under this distribution, which actually happens when the alternative hypothesis is true. Right? So let me just repeat once again. If the null hypothesis is true, the signal to noise will vary around 0. Right? If group 1 is upregulated, the signal to noise will vary somewhere in the positive values. So by how much it's shifted, well, it depends on by how much it's upregulated, right? So that if, in fact, the difference is not 0, the distribution will shift towards large positive values. And so now the probability of being outside of this goalpost, when in fact the protein is upregulated, is the statistical power. And beta is the type 2 error, sometimes people call, or so it's the probability of not detecting the change when in fact the change is present, right? So type 1 error is claiming a difference when there isn't one, and here is not finding the difference. And so what we want is to make the power as large as possible. So how do we make power as large as possible? Well, there are a couple ways. Well, first it's kind of trivial. Let's claim larger type 1 error, right? So now if we want to have larger type 1 error, of course, we will have larger power. So essentially, if we make more false positives, we will have more true positives, right? And so this is a trade-off. So the more conservative we are with false positives, the fewer true positives we find. The more we're willing to live with false positives, then the more true positives we have. But that's not very satisfactory, right? So there are other ways to do this better. And so a better way of so keeping alpha fixed, a better way of increasing the power is essentially moving the distribution further to the right. So we're talking about the signal to noise ratio, right? When protein one is in group one is upregulated. So how can we move this whole ratio of signal to noise to the right? Well, there are at least three ways. So one is, the larger the difference between the groups, the larger positive values the signal to noise will have, right? So if the protein is regulated very strongly, then this difference will have large positive values, right? And this whole distribution will shift quite a bit. But there are others. So if our variances in the populations are smaller, this will also shift the signal to noise ratio, right? Because we will divide by smaller numbers, so the ratio will be larger, so the whole distribution will shift as well. Meaning that if the proteins are highly regulated biologically and they have little variability, we will have a larger power to detect changes, right? So if they're tightly clustered around themselves. But here's the third. We also see the sample size as a denominator of denominators. So large sample size will also shift the entire distribution to the right. And this will increase this area under the curve. So essentially our goal is to design an experiment in a way that when the alternative hypothesis is true, the range of signal to noise is as large as possible. And this can be either focusing on proteins which are highly regulated, or reducing the technological variation, or increasing the sample size. So all of these three things will contribute to higher statistical power. And so now to answer your question about the sample size, so the way we actually think about this is like that. So we need to fix alpha. So what will be our decision region? We need to fix beta. So how much kind of, failing to detect the change we're willing to live with. Anticipated log-fold change. For example, below 2-fold change, I'm not interested, right? So you can kind of say, okay, these are the minimal fold change I'm interested in, and those are easy. And what we also need to know is an estimate of how much variation we have here. And this includes both biological and technological variation. And these are the hard quantities because they depend on the biological system, they depend on the measurement system, they depend on the stress, and so on. And so the first three you can usually fix just based on the common practice. This requires pilot studies and prior data to understand how much biological and technological variation we have. But if we know that, then essentially what we can do, we can say this is the variance associated with my signal, right? So this is my noise. I need to bound the noise, and again, there is a mathematical expression for the bound, and which involves alpha and beta, and you can essentially solve for n. But the upshot is that more biological replicates will, of course, increase the statistical power and therefore increase the sensitivity of detecting change. And usually what we do is we actually don't do it one calculation, we look at the range. And so you can make plots. For example, here, this is the fold change, and this is number of individuals per group. And once again, it will depend on your alpha, beta, and fold change, but most importantly, it will depend on how much variability you have. So, by the way, those are kind of the most challenging parts, kind of trying to understand how much variation we have. So on Wednesday, we will talk about the tools we develop, MSStats, and MSStats treats every data set that you analyze immediately as a pilot study for something else. And we will use this data set to estimate these variance components and to essentially make plots like that for subsequent use whenever you want to do a similar experiment with the same biological system and a similar technological kind of setting. Okay, any questions on that?
When you're doing power calculations, if you take your, like a data set you've collected previously, but perhaps from a different experiment, you know that, say, one sample group is going to be more different, can you do some sort of in silico modeling to represent what it would look like?
Sure. So essentially what we're talking about here is predicting the future, right? We say, okay, this is what my future experiment would look like. Well, how good are we at predicting the future, right? So the closer your pilot study is to what will actually happen, well, the better you will do because you have more information, right? Otherwise, you have to make assumptions or do sensitivity analysis, right? what if in my next study I will have a disease group which will have the variation, I don't know, twice as big as my current group, right? Well, you can build it in, right? You can, so that is easy to do, right? Or what if my fold change will be only half of the fold change that I actually see now? And so you can kind of play with this. So you need to have some rationale to specify these quantities, right? So if you have a direct insight, it's best, but otherwise you can just kind of play with this. So you can even take some variance components from your colleagues, right, or from publications and do that. But the further it's removed from your future study, well, the more difficult. And by the way, what can happen is, you have some glitch and your variances become worse than they were. That we cannot predict, right? So we can only work with what we have, right? So that's really, but sensitivity analysis is very common, and that's a really good thing to do. Yeah.
So just going back to the experimental design section, if your experiment has a lot of combinatorial constraints, so it's very difficult to identify a best design, how do you deal with that situation?
Well, you have the design under constraints, right? So clearly, you want to include the technological constraints in your design, right? The worst that can happen is that you cannot reduce the variance, right? So remember again, the blocking is all about reducing essentially the noise, right? So the more constraints you have, the less options you have to reduce the noise. So you may end up with noise which is larger than you would like, but you can always compensate for that by having more replicates. So essentially, for example, if you have a robot which can only handle like certain things, right? And you would like to allocate resources differently, but the robot wouldn't allow, you can always compensate for that by including more replicates.
What if that's not an option?
Okay, that's actually a very common question as well. So I like to say that my main job is to manage expectations. And so if you have constraints in terms of sample size and in terms of blocking and in terms of randomization, and you want to still proceed with the experiment, you totally can, right? There is nothing that prevents you. But be very clear about what is the reasonable expectation from the result, right? So if you have clearly not enough replicates and you clearly could not implement blocking in a way which reduces the noise, you will have to live with the fact that you will probably have more false positives and more false negatives than you would like. And at that point, it becomes kind of a business decision. Is it still worthwhile proceeding so that you can follow up on these things or not? So, there's no magic, right? So
I'm asking more about the question where consider the case where the study actually does have the right amount of power, but just coming up with a design that has the right blocking structure is really difficult because of like imbalanceness or something.
you can still, so, again, it's a combination of design and analysis, right? So you need to think about how the analysis would handle imbalanced blocking, right? And so there are certainly statistical methods which would be able to handle the imbalance. So remember in the beginning when I talked about blocking, I was looking at these pairs, right, and differences between the pairs. What if you have two healthy and one disease, right? So it's hard to draw a picture, but I can certainly do this with a statistical methodology. So it's actually a lot more flexible than it looks, right? You can really accommodate. There's price to pay in terms of the variance, right, in terms of your denominator. So suboptimal designs essentially will have a larger denominator. But out of multiple options, you can always find an option which minimizes that. And in fact, in more complicated, in industry, in more complicated experiments, what you would do, you would actually look for combinatorial search of the design space, which minimizes the denominator. And it may not be the smallest possible, but it will be smallest within your constraint space. If that's what it is, sometimes it's not necessary, but if it's complicated to that extent, you would just essentially do a search of all design space, which minimize the denominator. But most importantly, right, so especially if you don't have enough sample size, right, so managing expectations is really important, right? So what would be given my noise structure, the way I have it, right, so what would be the false positives, what will be false negatives? But that's, statistical experimental design is a whole area of research, right? And so then people think about like, what is the space, how to restrict the space, how to search through that, and so on. Yeah. Okay, so let's now talk about how bad p-values are, because now I want to make sure we finish in the last 15 minutes about things that, you know, I hope I convinced you that statistical analysis is important because it allows us this objective considerations of things, right? However, sometimes people attach too much importance to p-values, and, you know, they say, okay, the p-value has to be less than 0.05 or else, right? Or, you know, I can publish, and, you know, and so on. And this is a really wrong way to think about that. So let me just illustrate that. So this was an example also from Nature Methods from a different publication. It's a simulated study, so completely fake data generated on the computer. So they have two populations, A and B, fairly similar, and there's a difference of 0.05. So what they did here was they selected 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 replicates from population A and 10 replicates from population B. And they did the t-test, and they came up with the difference between the groups 146 and the p-value 0.005. Okay, we would reject the null hypothesis. But now they repeat it again. They again take from 10 points from here and 10 from here. And now they have p-value of 0.8. And the difference between the groups in a completely different direction. Repeat again, p-value again 0.85, and simulation 4 is borderline 0.09. So what this plot emphasizes is that the p-value is the result of the summary of the data, right? So we summarize the data to get the signal, summarize the data to get the noise, overlay it on some reference distribution, right? Because our data are random, our p-value is also a random variable, meaning that the p-value will not be reproduced exactly next time you repeat the experiment. And if we have a lot of noise in our data, so the denominator is large, it also means that the p-value will have a large variance. And so let's take a look at that. So now instead of doing it 4 times, they actually did it a large number, 1,000 times. And they make a histogram of p-values on the log scale just to emphasize the small p-values, because that's what we're interested in. So here they have sample size of 10. This corresponds to the p-value cutoff of 0.05. And you see that only 18% of the simulations had p-value less than the cutoff. The majority were quite large. They increased the sample size to reduce the noise, right? So now 46% were below the significance cutoff. Sample size 64, okay, now 80%. And only with sample size of 100, the majority of the p-values was in the significance range. So we should not be optimizing to get a particular p-value, because the p-value is a random variable. And in terms of the data analysis, we may select, you know, data processing and analysis steps in a way to get small p-value. It's a really bad idea. And here is another point which is related to that, and I think to me it's the most striking. So they do the same thing. So they repeat this simulation 1,000 times. The x-axis as before. So this is the p-value on the log scale. The y-axis is the estimate of difference between the groups. So they estimate here, and they just kind of make the bivariate plot. But most important thing is this histogram. So they report the difference between the groups, but only in the cases where the p-value was below 0.05. So essentially they report the fold change only for significant results of hypothesis testing. And so when the sample size is 10, what you see here is this histogram. And this is the true difference between the groups 0.5, meaning that everything, every reported fold change, when only reported among significant proteins, was way above the true value. So not only with small sample size, not only it's very rare that you claim significant result, but when you claim it, you grossly overestimate the extent of regulation. So it is way too optimistic. So the true regulation is much less than what it looks. And so when the sample size increases, so now the histogram kind of moves towards the bottom, more sample size moves to 64, and with 100 replicates only, now the histogram varies around the true value. So this is now a reasonable representation of the true regulation. So the sample size is really important also, not only just to gain sensitivity, but also to avoid this false optimism in terms of the extent of regulation.
So yeah, I love this slide. I've
What?
Well, you see there's a maximum on the y-axis on the right side of each plot of the
Here?
scatter plot. Yeah, what is that 68, 44, 32, 28?
That's as opposed to 0.5, right? So the y-axis is the difference between the groups, right? So here, the maximum, it reports 1.76 difference when the true difference is 0.5.
Yeah, but I don't know what the, so I know what all the numbers like the 0.5. What is 0.68 and why is it when at 100 replicates is reporting?
I think they took the mean or the median or something, you know. I think the labels of the y-axis are whatever the authors of this paper chose, but the y-axis is reflecting this. So oh, this one, yeah. So this one is the one that is sorry? Oh yeah, you're right. Yeah, this is the minimum, exactly. This is the minimum of this histogram. Thank you. Yeah, exactly. Yeah, this is the minimum of this histogram. So here, everything is above 0.5. Yeah, here, everything is above 0.44. Thank you. Here is everything about 0.32, right? And here is everything about 0.28. I didn't realize that. Yeah, but the important point is this, right? When the sample size is too small, not only we rarely find things, but when we find them, we're too optimistic.
So this is for pairwise comparison. What about when you start looking at multivariate statistics?
What do you mean by multivariate statistics?
So things where instead of looking at each individual protein, you start to look at, say, the network analysis and basically looking at all of the proteins rather than.
So what do you mean by looking at all the proteins?
So instead of doing your statistics just one by one per protein in your sample, you do statistics that include all of the proteins in the sample.
So my question was getting at what is the goal of analysis, right? So unsupervised pattern or hypothesis testing or machine learning, right? So remember that they have 3 different goals. So that's why I'm asking what by looking, right? So, okay, but you have a really good question because that's my next topic, and I hope we'll probably need to take a little bit over time if it's okay, maybe 5 minutes. This is exactly what I want to talk about next, and this is a really good question because everything I talked about so far, we look at one protein at a time. And here we're just going to keep collecting data over and over for the same protein, right, and see how much uncertainty we have. But in DIA, we clearly have more than one protein, right? And so how do we actually deal with that? And that's exactly the next topic. And so one of the pitfalls of that is the multiple testing. So now we have many proteins. Let's say we have 5,000 proteins in DIA, so we have 5,000 of those null hypothesis, right? And we have 5,000 p-values, and so what happens then? And here's an example also from Jeff Lick. Somebody had a poster making of an fMRI experiment on the brain of dead fish, and so they were looking for active regions. Pixels correspond to regions on the brain, and they were looking for activity, and they used hypothesis testing essentially at each pixel to see if there's activity or not. And they found many active brain regions in dead fish. Why, right? So because, well, if we look hard enough, we will end up finding something. Remember, we have this false positive rate of whatever, 5%, right? So 5% means that if we make 100 hypothesis tests, 5 will be, on average, false positives, right? So if now we have 1,000, we'll have 50. Right? And if we have 10,000, we will have 500 false positives, which are just by the property of this procedure, right? Should be present. So the large number of proteins will definitely inflate the overall proportion of false positives. So this is another way to look at it. Let's say we only have 2 proteins to make it simple, right? So we have protein 1 and protein 2. For protein 1, we have this distribution of values, right? And so we set aside the 2 tails, and this is 5%, right? And we will reject the null hypothesis and maybe make false positive with probability of 5%, regardless of what happens for protein 2. And therefore, our 5% is actually this area plus this area together. But now we do the same thing for protein 2, and so we reject the null hypothesis, and we have 5% here plus here. But the fact is that we have both proteins in the study. So in reality, we will make a false positive conclusion in any of this region. And of course, the combined area of the two proteins is more than 5%, right? Because now we have two proteins. And in fact, it's very easy to show that if the number of proteins increases, the probability of at least one false positive discovery is essentially 1. So we are guaranteed to make false positives. So how can we deal with that? Well, a natural way of dealing with that is to change our decision boundary and push it further back so that the area outside of this dashed line is whatever we want it to be, say 5% or 1%. So by pushing this decision boundary back, it means that we have to be more conservative. And this is the price that we pay for looking at more proteins. Essentially, each individual hypothesis test becomes more conservative. So we have to have a stronger signal to noise, go further in the tails to reject the null hypothesis. And that's really the situation. So there are many ways of doing this. So you probably are familiar with the Bonferroni correction for multiple comparison, where we take alpha and divide by the number of proteins. So this will be one way to push this boundary outside, and so that such that outside the probability is 5% or less. However, Bonferroni correction is too conservative, and if you have alpha 0.05 and you divide this by 5,000 proteins, well, it requires very, very strong evidence. So because we try to protect ourselves against false positives, and so the best way to not make false positives is to not find anything, right? So then for sure we will not have any false positives. But that's not why we're doing it, right? And so in 1995, there was really a breakthrough in kind of statistical thinking by saying, let us change our mindset. And let us, instead of trying to have a very, very small probability of making at least one false positive, let's say, okay, we can have some false positives. It's fine, provided that we can control the proportion of false positives in the list of our discoveries. And by doing this, we can be less conservative, so we can find more things. And so this was the change from false positive rate to false discovery rate. And you can, if we have more than one protein, so essentially we can have a table like this. So this is the truth. Number of proteins which do not change, number of proteins that change. It's not observable, we don't know what they are. What we do know is the number of proteins where we do not reject the null hypothesis and the number of proteins where we do reject the null hypothesis. So really the only observable number is R here. And Bonferroni will control the probability that V is anything by 0 and will set this probability very small. So that's why it's so conservative. False discovery rate instead says, okay, we can have V different from 0. We want to control the average proportion of V divided by R. And there is some technicality that we don't want to divide by 0, but in these experiments we always find something, so it doesn't really matter. So we want to, if we kept collecting data and repeating the procedures over and over, we want to control the average proportion of V divided by R. And unfortunately, I don't have time to talk about how this procedure works, but what we can do is to adjust the p-value such that the control cutoff of the adjusted p-value, let's say 0.05, corresponds to false discovery rate of 0.05. And so what people usually do, they would make these volcano plots where the x-axis is the log-fold change. This is what we call practical significance. And the y-axis is the p-value adjusted to control for false discovery rate on the negative log scale such that large values correspond to statistical significance. And we will use the adjusted p-value cutoff here for statistical significance. We can also use additional fold change cutoff for practical significance. And so those corners will be the proteins we're interested in. And so this would be the type of plot that MS Stats reports. And so now, just very few comments going more into the fact that we have more than one protein. And I think this is particularly important in DIA experiments because we have the opportunity to have really deep proteome coverage. And so how does this play out together with statistical analysis? So the first thing is the sample size. And now going back to your comment. So just as before where we could work out the boundary for the variance in a single protein case, we can work out the sample size calculation when controlling for false discovery rate. And so MStats would produce plots like that, and again, it's done completely analytically based on the variance components from the pilot study. This was the plot we had before, where for a fold change we have number of replicates, and this is just one protein in the study, which doesn't really happen. And now the two lines here, this will be the sample size if we have multiple proteins. And the first thing you see this, like these lines are above this line, right? So if we have multiple proteins, we have to be more conservative. And therefore, we have a larger sample size to keep the same power, right? But what people often overlook, and I think this is particularly relevant for DIA experiments, is the impact of how informative our proteins are. So here, the bottom line, the middle line here, is the sample size needed if we only expect half of the proteins in the study. Well, if we expect half of the proteins in the study to be differentially abundant, and the top line is the sample size needed if we only expect 1% of proteins to be changing. And in DIA experiments, this is often the case. So essentially, if few proteins change, we have a deep protein coverage, but only few proteins are regulated, we need a larger sample size. And the intuition is that, again, we're trying to protect ourselves against false positives. If everything is changing, right, and everything is a true change, we have less opportunity for false positives, so we don't need to work as hard. If we have 10,000 proteins and only 1% is changing, we have a lot of opportunity for false positives. And therefore, we need to work harder to protect ourselves against false positives. And work harder in this context means larger sample size. And so now for sample size calculation, we will also need like fold change alpha, fold discovery rate, variance components, but we will also need an estimate of what is the percentage of proteins we expect to be truly changing. And again, we can have a sensitivity analysis. And the smaller number of proteins are changing, the larger sample size we need. So this is really the importance of the sample size. And so now I just want to finish because with one more consideration. So oftentimes people say, well, you know, but then downstream we can do network analysis, we can do like machine learning methods, you know, and so on. And all of this can kind of compensate for some of the false positives and so on. So I want to finish by changing the discussion a little bit and focusing on the third goal of statistical analysis, which is biomarker discovery, right, or classification, where we have machine learning methods which predict the status of individual subjects and show that for this type or any other analysis, we essentially have the same issue, that if we have a deeper protein coverage and more proteins in the study, we need to have a larger sample size. So let me just illustrate this for you. So this was an example. So this is an SRM experiment, but it doesn't matter. So we will have the same issues everywhere. So we have the objective is to classify each subject into two groups. So this was a study of colorectal cancer versus control. So, and we would use random forest, just one of the machine learning methods, to determine this type of boundaries between healthy and disease. And the reason why we took this example was because it has a large number of replicates. It had 200 subjects in the training set, so with 70 proteins. But then there was also a second follow-up experiment with 269 subjects, which was a validation set, where whatever boundary was determined here was essentially applied to these new subjects to see how it works. And so what we decided to do is this. We wanted to understand how the sample size versus the number of proteins plays out in terms of the ability to find those boundaries, right, and find them accurately. And the idea is that if the boundary is accurate, then, you know, you have a few more subjects, a few less subjects. It shouldn't matter, right, because the boundary should be resistant to that. And so what we did is we took this training cohort and we started randomly selecting a subset of replicates from this of various size. And then we also took a different subset of proteins, 10 most abundant proteins, you know, 20, and so on. And for each subset of proteins and subjects, we applied a random forest classifier, And we recorded two things. We recorded the accuracy on the validation set, so how well this boundary performed. And we also took the 10 proteins which are most selected as most predictive by the random forest. So this will be our 10 most promising biomarker proteins, right? And we did it over and over 100 times. And so here we could record the average accuracy. So it's the proportion of true positives and true negatives among all the subjects. And we also made a plot like this where these were the top most frequently selected biomarkers, and this is how often they were selected in 100 iterations. So ideally, we want these numbers to be close to 100, right? So that they are repeatedly selected over and over. So that's the idea. So let me now show you what actually happened. So first we took the training set, and we only said, okay, let's pretend our study only has 10 replicates. So we'd over and over sample 10 subjects from the training set, and we took only 20 proteins which are most abundant. So let's say we have such a limiting technology, we can only quantify 20 most abundant proteins. And so select 10 subjects, select 20 proteins, apply random forest, rank proteins, calculate the accuracy, do it again, right? So we see that, first of all, the average predictive accuracy was 0.49, right? And the proteins were selected at most 50% of times as biomarkers, meaning that out of 100 repetitions, at best, half of the time we selected the same protein. Otherwise, some other proteins were selected, meaning that this choice of the predictive proteins was not very stable, because half of the time we would have some other proteins showing up as predictive. Now, if we increase this to 40 proteins, so we go a little bit lower in the dynamic range, well, the predictive accuracy improved, 0.52. Now we have more proteins to be predictive, but look at this. The number of times the proteins are selected went down. So we are now having different proteins coming in and out depending on which subjects are in the study. And now we took more, took 70 proteins. Look at that, the predictive accuracy went down. And this is because when they increase the space of possible predictors, with 10 subjects, the method has a hard time distinguishing systematic boundary from random artifacts. And so more proteins with the same sample size reduced the accuracy, and it reduced the kind of how systematic we can select the same protein over and over as a biomarker. And then we did this kind of by increasing the number of replicates. And it is really striking that across the board, if you increase the number of proteins, the accuracy first goes up and then goes down. More subjects, first goes up, then goes down. 100 subjects, first goes up and then goes down. And it's only with the full 200 subjects that the predictive accuracy is much better than before, and it didn't really go down as much with the number of proteins. And you also see that with 200 subjects, the stability of how often the same proteins are selected over and over with different subjects became higher. So this illustrates that the large protein space introduces challenges, and this is, by the way, not new at all. In statistics and machine learning, this is a well-known phenomenon. It's called curse of dimensionality. And the only way to compensate for the increasing protein space is to increase number of biological replicates. And what we're finding is that we can also use this type of simulations to predict sample size for classification and not just for hypothesis testing. And in fact, it's implemented in MS Stats. And so now we can try to project different things. So here we have, based on these variance components, we look at the number of proteins, we look at the number of subjects, and here we plot the average proportion when each protein is selected. Each truly predictive protein was selected as a biomarker. And we see that so when we have large number of proteins, few subjects, it's really abysmal, right? And so the only principled way to consistently select correct biomarkers is to increase the sample size. And so then we can do this in many ways. We use different data sets for variance components. We use different kind of projections, predictive proteins among high abundant or among low abundant, and so on. And these numbers vary, but qualitatively you always see the same pattern, that as the number of proteins increases, we need to have a much larger sample size than what people typically do currently if we want to consistently select the same proteins as predictive. So the point is, and this is going back to the experimental design, when the protein coverage expands, the price for this is the need for a much larger number of biological replicates than is typically done. And by the way, just to finish very last slide, to show that even for prediction, the principles of experimental design hold. So here we took another data set. This was the colorectal cancer versus controls from TCGA, but it was kind of combined data set from multiple sources. And so what you see here is, so we wanted to look at this because it has much larger number of proteins, 3,000, relatively large number of replicates, 100 replicates, right? And we did the same thing. And so on one hand, you see that you have the same problem. The proteins are not consistently selected at all, right? Especially when this protein number increases. But look at this. Predictive accuracy is 100%. No matter what we do, predictive accuracy is 100%. And so what turned out, it's the same problem as before, so that the cancer subjects were collected by a different lab than the controls. And so no matter that every protein here is predictive, but it's predictive of the lab and not predictive of the disease, right? So the flaws in experimental design carry themselves over, not just like for hypothesis testing, but for any other type of analysis. Okay, this is the end. So just some takeaways. So I think some things we talked about and others will be for Wednesday, but just to summarize, first thing is define the problem, right? So we need to say, not just looking at things, right, or not using like signatures or some other things, but we need to be very specific about how we translate biological goal into statistical goal. Experimental design, really the purpose is to avoid bias and inefficiency, and the tools are randomization, replication, and blocking, and prioritize biological replication as opposed to proteome coverage. And if we go for proteome coverage, then we need to make sure that the biological replication follows appropriately. And these are things which I will kind of go back to on Wednesday. It is important to follow a predefined design and analysis protocol. Do not alter the design, not cherry pick the data parameters, because we can overfit essentially and show anything we want. And if we do have constraints, we need to understand and state limitations. And in terms of reproducibility, we need to document all the steps, and in our experience, your code is the best documentation. So your scripts you use for analysis of data can be used later for others to repeat or reproduce your results. And a lot of the figures from the presentation are actually coming from this series of points of significance. So there's a lot more there than I could show, so I encourage you to take a look at those. Sorry for going over time.
So we can maybe take one or two quick questions if there are any. Just to remind people to use the microphone as well. That'd be great. It doesn't actually make any noise, but it would help us for capturing the audio. Otherwise, Olga will be back on Wednesday.
And I will be here in case you just want to talk to me offline. You're most welcome to.
Okay, yeah, one.
So in your simulation study, you assume that the biomarkers are uniformly distributed across the expression range.
No, so that's kind of what we tried to, you can really vary this in any way. So these are predictive in high, this is predictive, kind of uniform, this is predictive among low abundance. And you see that for predictive and low abundance, the numbers are lower, right, than in predictive and high. So yeah, and you can do this type of sensitivity analysis for different things, right? The important point here was that the large values on the x-axis always correspond to to bad prediction, unless you have large sample size.